Hello, everyone, and welcome to Lab Manager's Ask the Expert webinar series. My name is Mary Beth Didana, and I'll be moderating this discussion, Accelerating Protein Characterization with Mass Photometry. We like our webinars to be very interactive, so we encourage you to submit your questions to us at any point during this webinar. Our speaker will begin with an overview of the topic, and he will then be joined by Lab Manager's Editorial Director, Scott Hanton, for an informative discussion on mass photometry. Our speakers will address your questions and comments throughout this webinar. To ask a question or leave a comment, simply type your query into the Q&A box located on the right-hand side of your screen. We'll try to address as many questions as possible during our time together, but if we happen to run out of time, I will forward any unanswered questions to our speaker and he can respond to you directly if possible. Please visit the Handouts tab on the right-hand side of your screen for additional information about this presentation. I would like to remind you that this webinar recording will be available on demand shortly following this presentation, so please watch for an email from Lab Manager on how to access this free video once it's available. I would like to thank our sponsor, Refeyn, for their support, which allows Lab Manager to offer these webinars free of charge to our readers. So with that, I'd like to introduce our presenter for today's webinar. Dr. Tomás de Garay is Product Manager with Refeyn. Tomás, thanks for joining us.
Hi, thank you, Mary Beth, for the introduction. Yeah, I'm very excited to be joining you today for this webinar. It's called webinar, but really more meant to be a Q&A, very extended session. So I will just have today 5 minutes introduction to make sure everybody's on the same page. The ones of you who might not have heard about mass photometry before or might be wondering what this is, I'll give an introduction on the technology, but really this is meant to be a discussion session, so feel free to submit any questions as I go, and we'll have a very nice discussion session afterwards. So today we're going to be talking a lot about biophysical characterization of biomolecules, proteins, nucleic acids, RNA, DNA, even things like viral vectors, this kind of stuff. That's where mass photometry sits. But basically, if you look at the screen, all of these questions at the moment on the screen are things that we can look at in different ways. And that is because ultimately they all imply a change in mass of the biomolecules, and therefore a mass readout can be very helpful into answering these questions. So anywhere from sample characterization as to purity of a certain protein sample, aggregation, degradation, integrity, all the way up to the formation of complexes, stoichiometries, what the stoichiometries are certain interactions, are interactions forming or not under different conditions. All these things can be answered through a single readout, which is molecular mass or complex mass or particle mass, right? That's essentially what mass photometry measures. Mass photometry is a single molecule or single particle mass measurement that happens thanks to light scattering. And this is the range of biomolecules that we typically work with, from the 30 kilodalton on the small end up to 5, even 6 megadaltons to some instruments is things that we can work with. So just a very quick intro on how exactly this happens. So if you haven't really seen a mass photometry instrument before, or a file or a measurement, this becomes a little bit real in your head. Very simply, what we are doing is we're shining light onto the biomolecules, and we are measuring the way these biomolecules interact with light. And in particular, how they scatter light or how much light they scatter. So if we shine light onto a molecule of low mass, we will get a low intensity signal, whereas a molecule of higher mass will get a higher intensity signal. And it's really as simple as that. So this is how a measurement might look like. You know, when you look at the computer in the lab, you'll immediately see something like this. Each one of these dots, we call them events on the image, corresponds to a single biomolecule. In this case, could be a protein complex. And the intensity, so how dark they are in the center, is directly proportional to the mass. Therefore, say this one here, which is dark, is high intensity, would be a higher mass, versus this molecule here, that would be a lower mass because the intensity is lower. And that is what gets quantified. It's literally the intensity of those pixels in the image. So it's a very direct measurement. Now, we are not quantifying just 6 or 7 like in this one frame. We take a 1 minute measurement, and we will quantify a few hundreds to a few thousand of biomolecules to get to the final result. So here, well, there is an example, but I'll try to show you real data, which is always a bit more descriptive. This is an antibody, and this is an example of how a typical mass photometry measurement can look like after that 1 minute of recording. What we are having here is a graph with mass in the x-axis, so kilodaltons, versus number of counts, so number of individual molecules counted in the y-axis. So the typical measurement would be the one on the top, right, where we pull all of the thousands of molecules into a histogram. In this case, we have a monomeric antibody at 150 kilodaltons, but we also have aggregation of this antibody. There is a dimer at 300 kilodaltons, a trimer at 450. The lower part, I've just put it for this example so that you can see all the individual molecules that were quantified that gave rise to this histogram. So if you look even around here, it's hard to see on the histogram because you would need to zoom in a lot, but there are even a few molecules on the tetrameric area, and those are real molecules. They are counted, right? So whether they are relevant or not, that's a different question, and we can get on that during the Q&A, depending on your application, but they are real. So we get a mass value, and we also get, just by counting the number of molecules, a relative abundance, a proportion of each population. So I'd like to show here, and this is literally the last slide, so I'm hoping for a very interesting Q&A, how this looks in real life, right? If we play this video, this is a mass photometry instrument, and that is the area where the sample gets loaded. So that's a colleague of mine pipetting that's 10 microliters of very low concentrated protein sample. And it's literally from a tube in any buffer. There is no sample prep. As soon as you close the lid of the system, you look on the screen, each one of those dots that you're seeing there is in real life one molecule that is being measured at that moment in time. And this will be recorded for 1 minute. So it's as simple as that. That is mass photometry in a nutshell, so to say. And with that, I'm very much looking forward to see what the audience wants to talk about today. So we'll direct the discussion in the direction that anybody wants.
Thank you, Tomás. That was a really interesting introduction to mass photometry. I enjoyed it, learned a few things. I think for this conversation today, we ought to start out with the instrument and the science. So let's start there. You've talked about some different things that mass photometry can do. Where does mass photometry fit in a typical biochemistry workflow?
Yeah, definitely. That's a great question, and it's good and bad because it's not so easy to answer because it can fit in many places, which is one of our strengths. It's quite versatile, but we typically see it in one or of two settings, or sometimes in both at the same time with the same instrument. It can either fit upstream of something else, a downstream experiment that you want to do with purified proteins, purified nucleic acids. That can be a structural biology assay, say cryo-electron microscopy. It can be an interaction assay. It can be a functional assay with an enzyme, something like that. So mass photometry can sit upstream of all those assays in I would like to call it even like kind of a QC or quality check of the sample. Basically, every time you're going to perform something else that might be quite time-consuming, rather expensive or involved technique, you want to make sure every sample that you're bringing down to those techniques is the best sample that you can bring and is in the state that it's supposed to be, so you don't get surprises and the results are easy to interpret. If you just take really 1 microliter of your sample, of your aliquot, you pop it in the mass photometer, you will get this single molecule, very powerful information that will tell you whether the protein you're about to use is still the same as it was a few weeks ago when it was purified, maybe by somebody else, maybe in a different place, or has it aggregated? Has it degraded? Was it a complex and now it's no longer a complex? That can seem a bit simple, but it's very powerful and can save a lot of time in downstream analytics or techniques in any way. The other place in which it can sit is as the main experiment itself. So you can imagine mass photometry as being the main figure on a paper or something like that. You can use mass photometry for the analysis of interactions, and we can delve more on that later as well as to how that fits with other interaction techniques or assays. The analysis of the formation of complexes, maybe over time, maybe over different conditions, different buffers, different mutants, different clones, things like that. And you can really get to analyze all of those parameters. So what's the status of the sample and get readouts on them. So either upstream checking before something else, or as a main analytical tool for studying a certain biological process or a certain interaction are the two biggest things. The good side of that is that it's so fast to use that you can possibly do two at the same time, or some people in the lab might be using it for one thing, some people might be using it for a different one.
You mentioned in your presentation that you could look at things down to 30 kilodaltons. Is 30 kilodaltons the lowest mass that you can quantitatively detect?
Yes, 30 kilodaltons would be the lowest mass, and then up to 55 megadaltons. Yeah.
And what's driving that lowest mass? Is it how the light interacts with the particle, or is it something about how the instrument functions?
It's important to say it's not a theoretical limitation, so to say, of the physics behind the technique. It's more of a technical limitation. So it's ultimately the biggest factor is shot noise, and therefore signal to noise ratio. So with the noise that we are typically getting, then the kind of safest specification for us to give is 30 kilodaltons. We are sure a protein of 30 kilodaltons scatters enough light that it will be consistently above the level of noise that we can expect given the setup that we have. There has been a lot of work into optimizing that. So it used to be 40 kilodaltons a few years ago, and in the original versions, it was even more like 50 or so. But it's really about that. That's not to say that some people sometimes in some setups are able to see a little bit lower molecules. It's not a very, very hard limit, but that's the one we're comfortable with saying everybody should be able to do it. And we test all our instruments for 30 kilodalton protein.
Great. That explanation was really helpful. So you mentioned that lowest range is due to background noise. How sensitive is the instrument or the technology to vibrational or acoustic noise?
It depends what you compare it with, but I would say somewhere mildly sensitive to vibration. So yes, vibration is definitely a factor, and that's a very good catch. You would need to think where you place the instrument in your lab. We recommend not placing it close to sources of big vibration, like just not put it on top of an ultracentrifuge or next to a -80 fridge, something like that. Other than that, it's okay in most labs. And that is because it's not as sensitive to vibration as, say, a single molecule microscope, for instance, or something like that. Those are much more sensitive. It's also, there is a small device just below the mass photometer that actively cancels the vibrations in the environment. So with this device, we can really control for most environmental factors. So I think bottom line is, as long as nobody's literally tapping on the instrument on top or slamming the door next to it, it can go on most labs.
So it doesn't need to be next to the cryo-EM in the sub-basement. It just needs to be carefully placed wherever you are.
Yeah, exactly. No, definitely. No. You can save the precious basement space for what the things that actually need it and will benefit from it the most. Yeah.
Perfect. Now you've talked about what the lowest mass is that can be detected. What about the smallest mass difference? between two different analytes in a solution?
Yes, definitely. It depends a little bit where you are on the mass range and what are the relative proportions of the things that you're analyzing. But as a rule of thumb, we are comfortable saying you can be able to see differences of roughly 15% of the absolute mass value of the molecule. It will be a little bit more in terms of percentage, more like 20% towards the smaller end of the mass, towards the 30 kilodaltons, and a little bit less towards the high end because it's not fully linear, and that's because one of the elements is the short noise that we mentioned before, and that is constant throughout the range, whereas another element is proportional to the mass of the particle. So essentially, any macromolecular interaction, so to say, or macromolecular differences, say protein-protein, nucleic acid, anything like that, are things that we see on a daily basis, basically. Anything that goes more into the 2-3 kilodaltons, small molecules, or hundreds of daltons, or oligos, things like that, that would be too small.
Excellent. Thank you. When you say that you can distinguish 15%, is that like you showed those peaks before? Would that be a shoulder on the peak, or would there be some resolution between the peaks at 15%?
Yeah, it could be both. It will depend a little bit on the sample. I think the first point to mention is depends on the homogeneity of the sample, so how monodispersed your sample is, right? The best case being a purely monodispersed recombinant protein that will have the best resolution, as in the peak that you see will be the narrowest, right? Mm-hmm. If there is some heterogeneity inherent to the sample, obviously that will affect things like glycosylations or small degradations, things like that. But assuming sample monodispersity, the way we are stating resolution in numbers is for BSA, which is a 66 kilodalton protein, the width of the peak at half the maximum, so how wide the peak is at half the height, is 25 kilodaltons. So you can imagine if you have BSA at 66 kilodaltons plus or minus 25 kilodaltons, you will see two peaks that overlap up to the half the height of the peaks. So you will have two clear tops. You will be able to separate the two populations, but there will be some degree of overlapping. That's 25 kilodaltons of width for BSA at 66 kilodaltons. It's 60 kilodaltons of width for 10 times the mass of 660 kilodaltons. So you can see how it's not linearly scaling with mass. It will really then depend on what's the relative abundance of the populations you're looking at. Obviously, if they are, say, 50-50%, that's the best case scenario in which you will be able to see two peaks. And even if they overlap a little bit more, it will be easier to see the two tops. Whereas if one of the populations is lower, then you will start to see it as a shoulder, and that shoulder will stop to be a clearly differentiated shoulder at some point. That will depend on the relative abundance of it. So that's why it's a little bit hard to give a number, but somewhere on that range, hopefully the 25 kilodaltons for BSA helps a little bit to people to imagine things. So definitely things like monomer dimer is something that we see fully resolved down to the baseline in every case.
And this should be familiar to bioanalytical chemists, right? Because we see the same effects in chromatography or mass spectrometry. And so it's not a unique or unusual case with mass photometry.
Mm-hmm. Yeah, definitely. I think the only thing to mention there is in this case, it's really the only factor that affects that is the mass of the particles. So any mass change is what will dictate the particles moving on the x-axis. Any mass heterogeneity is what will dictate the peaks getting wider or the populations getting wider. It's not really related to the shape or the size of the particle or the mobility on a column or any other factors. It's literally just the mass.
Thank you for that clarification. I think that's useful. When we're talking about using the instrument itself, What kind of training or expertise is required to get good data?
Well, that's an easy one. Very little. I mean, you've seen it from the video, right? It's obviously that video is a little bit cut and the analysis is not shown, but you pipette the sample on the stage. There is no sample preparation needed other than getting to the right concentration. So you might need to dilute the protein. You find focus, which is done by the computer, and then you click record. And you might be talking 3 minutes from pipetting to getting the final graph in the analysis software. So our training is just to give you an idea. When our training engineers go to on site, they tend to last about 3 hours roughly. And that's to train a group of 4 or 5 people to a standard user level. So it's the kind of instrument that everybody can be trained in, and people typically can be independent on the use of it, even if they're sporadic users.
So it's not something where you're going to expect that the average user is a PhD with experience. This is a tool that can be used broadly across the lab staff.
Yeah, not necessarily. You don't need to be an expert on it. You don't need to spend months or years on it to really interpret the data or be able to tweak that tiny setting that is hidden behind the menu. But if you tweak it, everything gets better.
Let's talk about the system itself. How are systems typically managed by laboratories?
That would depend a lot on the laboratory setup, but I would say because of the ease of use and the short time that it takes per measurement, is the kind of system that's very popular in shared spaces like platforms, like analytical services, things like that. Anybody can get trained and they can use it for a few hours, get the results on a very regular basis. So they tend to be a lot of, multifunction device, because one person here might be using it for one thing, one person there might be using it for a different application. It's very versatile, and therefore it makes sense to be in such environments. There, we see a lot of these places where they have what some people call a super user or a manager or a head or something like that, which is the person in charge of the system, in charge of training other users, or the more tricky applications, or the I would call them funny applications. So the things that are not out of the box. There are people doing very, very interesting things out there with customized analysis, particle tracking, things like that. But as much as I love to talk about those and to see how people use them, they are not very common, because most things you can really do out of the box in our software.
Great, thank you. What sort of maintenance does the system need?
Maintenance is also same as training, quite straightforward. Obviously, we do have a service team and we do provide annual service visits. It's mostly not because there are components that need very regular changing, or movable parts, or lasers that need to be replaced every 6 months. There's nothing like that inside the system, but we do want to provide all the users with the reassurance that their systems are up to specifications. So once a year we go, we visit them, we make sure they're working fine. If something needs to be adjusted, it gets adjusted. But in terms of everyday usage, there's really one thing that I advise to everybody who has a system or is thinking about setting up a system somewhere, is clean the objective and make sure all the users clean the objective. That's the one and pretty much only thing that you need to worry about as a user. It takes 5 minutes. It's very easy, but it's still an optical device with an objective, and that is the only part that you need to really take care of. As long as everybody does that, there's not much else to be done around it, really.
I think that cleaning is an important point. Mass photometry can be very sensitive. Carryover can lead to misleading results. So how often does the instrument need to be cleaned, and how much does that depend on the various different users who are using it?
Yeah, definitely. It is quite sensitive. We're still talking about a single molecule technique, indeed. And it's a benchtop device that you see there. You go pipette the sample. It's not in the basement. It's not. It doesn't look like such a powerful thing. So people sometimes forget about it, which is probably a good thing. It means it's simple to use, but it's still a single molecule device. So the cleaning of the objective, it's something that takes 5 minutes, really. I mean, even less. But we recommend to do it for sure at the end of your session, your experimental session, so that it doesn't stay like that overnight or anything like that. If you are measuring for an extended period of time, say maybe every couple of hours or so, it's good to keep it that clean to the objective. So you just, once you are removing your slide, your consumable from one measurement to another, you can take that chance to clean the objective a little. Other than that, there is not much else. So there is not really a chance for cross-contamination of samples, so to say, because I mean, if you could see on the video, the samples get pipetted onto a very small well. That's a silicone cassette on top of a glass slide. It looks like a microscopy cover slip slide, a little bit longer, with 6 wells on top. You will have 6 measurements on that consumable, so you can pipette 1 sample per well. You do 1 measurement on there, and then that is discarded. It's just a glass piece, so it's really inexpensive. And you put another one for the next 6 measurements. So you don't need to clean those. You don't need to, it will honestly be very hard to clean, but they are so inexpensive. Those are disposable, and the samples are isolated from each other.
That's a good clarification. I hadn't realized that from the video. I was worried about cleaning between every sample. So that's a nice innovation. Tomás, can mass photometry, the way you're doing it, can it be automated?
Yes, it can. Yeah, also. I mean, that was one of the first questions when we first launched the first ever mass photometer quite a few years ago now. So we work on that quite hard, and at the moment it is possible. So what you have seen in the video is obviously the manual version where a person was pipetting onto the system. There is indeed also an automated version. So what we're doing is, instead of pipetting manually, we are pipetting with a robot on top. Let me see if I can go back to the slides. To sharing the screen, I can probably show you a picture of what that looks like. There we go. This is the automated version. So what we're having is, instead of the person pipetting, we're having this area where it's hard to see, but that's where the consumable is. So that's where the person was pipetting before. The mass photometer itself will be down here. And then there is a robot that will take the samples, will pipette them automatically. This can do a run of 24 samples in about 90 minutes. That's completely operator free. So it's saving up a lot of time from you as an operator, as a user, but it's also removing the chance for user error or for user-to-user variability, all these kind of things.
Oh, that's great. And that's a cool application. And in my head, I had what your first video was, right? The individual pipettes. It's nice to hear that, especially for people who have more complex or broader research, that they can include it in their automated workflows. Let's shift the conversation now to some applications.
Mm-hmm.
Can macromolecular protein-protein interactions be assessed with this tool?
Yes, yeah, for sure. I think that would be, again, into this second category that I mentioned of where does it fit within the biochemical workflow, as in, it's not just a QC tool or an enabler tool. It can also be the main experiment, and it can get up to a complex experiment. I would say mass photometry is particularly well suited for interactions that get a little bit complex, right? So if you have a protein A plus protein B equals complex AB, and that's it, there are many ways that you can analyze that depending on what kind of information you want to get out exactly. But As soon as that gets a little bit more complex, say you have A plus B equals AB, but you also have A2B2, and maybe there is also A2B1. There are multiple stoichiometries that coexist, so there are multiple equilibria that coexist at the same time. It can be quite hard to interpret the results of techniques that work in an ensemble basis. So in which what you get is basically an average measurement of all the molecules in the solution. And that is really where mass photometry can add a lot of value and where we see a lot of people using mass photometry for. Again, maybe I can show you one example if I go back to the presentation in here. This is, to switch from the only protein world, this is a protein DNA interaction now, but it's a very good example. And you can think of this system as any kind of molecules you would like to put together into a well and assess whether they interact or not. What we have here, we did this in London at the Crick Institute, is a nucleosome formation experiment, right? So we first have DNA alone, and this is just a control, right? We have, again, this graph that was kilodalton in here, so mass versus number of molecules, and we see there's pretty much a single population in here, right? And then we start adding the histones, and we wanted to assess how was the nucleosome formation and how many nucleosomes could be formed. So in the first experiment, we did a 1.7 ratio of histones to DNA. And you can see how there are now two new populations there. One that if you add the masses, it would correspond to the mass of the DNA we saw here, plus the mass of the histones of one nucleosome, right? And then another population that corresponds to our two nucleosome population regarding the masses, right? So you could ask the software how many molecules are in this population versus how many are in this population. This is already two different reactions, right? Two different equilibria that are coexisting at the same time. But if we increase the ratio of histones a little bit, that's the latest graph here at the 2 ratio, we can now see there is a new population emerging, and the mass of this population would correspond to 3 nucleosomes plus the DNA. And you can see that one nucleosome population is reduced now in relative compared to the other two, right? And if we keep pushing, we keep kind of displacing the reaction to the right, now adding 2.3 ratio of histones to DNA, we see the 3 nucleosome population has now increased, and this is quantitative, right? We could get a percentage out of here. So you can really see how these reactions in plural, they coexist at the same time in the same tube, and they move as you change the parameters. Interestingly, you can also see how there is not another population coming up. There is not a 4th nucleosome population in here, which in this case makes complete sense because the length of the DNA, if you look at the number of bases, it would only allow for 3 nucleosomes. So there's no physical space for a 4th nucleosome. But in other cases, it might not be the case. So with a single measurement, you can get all this information of this, in this case, 3 different reactions happening at the same time. You can get the information of what the stoichiometries are possible and which ones are not, which ones happens in your specific condition, in that buffer, in that moment, at that conditions. And you can also get quantitative information on the ratios of the different populations. And then you can imagine applying this to anything. You can imagine applying this as to how it changes over time and doing a time course, how it changes in different salt conditions, how it changes if I change the DNA sequence, for instance, in here. Hopefully, it shouldn't change too much for this experiment, but it could for others. So I think that's, yeah, sorry, go ahead.
One thing I notice on the slide that you're showing right now is that lowest mass peak. We see it in when you've added the histones, but you've put a red star on it in the 2.3. So why are you calling that peak out? What is it?
Yeah, this is, oh, sorry, it's not called on the slide. It was too small. But this is the free histones. So at some point, because we're adding too many to a certain extent, so there is another peak of free protein in here only. It's a bit of a funny one because histones are not completely soluble, so it's probably not representative of the full histone population, and it has to do with the process that nucleosomes are assembled themselves. But it's a very nice catch indeed, because that's also something you can see. You can compare your individual populations, your individual species, and the complexes, and whether you are saturating a certain interaction or not, or whether you still have some of the unbound species, in this case, or whether you don't have any unbound species, or very little, like in this case here, for instance.
It might not be appropriate for this experiment, but is there also a time component? If you said that the measurement takes about a minute, but if I left it in the well and came back at some time later, could I measure changes in time, basically getting some proxy for kinetics as these different interactions occur?
Yes, you could. I think the best way to do that would be by incubating the sample in a tube, in the Eppendorf tube, in whatever conditions you want to incubate it, which could also be at, say, 37 degrees maybe, or something like that. And then every 5 minutes, 10 minutes, every hour, every day, taking a small liquid of that sample and then measuring it, and then discarding it, because after the measurement, there might be some evaporation in the well. We're talking about a 10 microliters volume that we're using for measuring. So it's really, really a small amount. And it would be best to change the well. But we have done that. And yeah, you can definitely see changes in time. Actually, I think in the thing that we sent for the handouts, so in the handouts, there is one application note. And one part of that application note is actually that it's a time course of a reaction that happens over the course of 3 hours. We didn't know this when we started the experiment, right? But that was the whole point of the experiment, was to see how long it took for that reaction to reach equilibrium, which was unknown, because a lot of experiments downstream were depending on that. So we needed to establish a protocol as to how to dilute this protein, wait for a certain time until the reaction reached equilibrium. This was an oligomerization reaction. And then after that, perform other experiments down the line. And we wanted to measure how long that took, and depending on different buffer conditions as well. So that's a very nice use case that's in the handout in the form of that application note, definitely.
And thank you for the reminder to the people participating in this webinar. There are 2 handouts out there. I encourage you to go have a look at them. One of them is the application note that Tomás just mentioned. The other is a brochure for the instrument itself. So if you want more of the technical details about the instrument, that'll be there in the brochure, and they're both in the handout section. I encourage you to go download both of those. Tomás, when you showed the slide with the protein-DNA interaction, you talked about the results being quantitative. How quantitative are they?
Yeah, they are quantitative in 2 ways. The most obvious one is we're quantifying the mass of each population, right? That's the mass photometry. That's what we measure. That's the main thing, the main outcome. So you will get this straight from the analysis software. The software will feed function to the population to account for the small deviations, the error in the experiment, and it will provide the median of that function being the mass of the population. So you get a kilodalton number, basically, for each population. The second way it's quantitative is in the relative abundances of each of the populations that you're looking at, or each of the areas that you want to define on the graph, right? So if you have clearly defined populations, like in the example I just showed before for the interaction, where there were multiple peaks that you could see on the screen, then the software will also feed a function to all those peaks and will provide a number, as in, under this peak there is this many molecules that were counted. And that accounts for a certain percentage of the overall number of molecules that were measured across the whole range of masses, right? So you do get that number. And that is calculated as simply as that. Is by counting molecules, because we are counting individual molecules and then plotting them together. But we have to remember that graph, that histogram, is the result of plotting together 2,000, 3,000, 4,000 individual molecules. So we're just counting, there were 350 here, there were 483 here. What's the proportion of that? So it's a very direct measurement in both ways, the mass and the proportion of molecules in there.
So when we think about the proportion of molecules, it's counting the number of molecules that are detected. So each peak is measured by how many events occurred. But is there any impact of the weight of what has occurred, or is it simply it's a particle and it's been counted?
Now, it's one particle, one event. So we don't have a bias for larger mass particles or lower mass particles or something like that. If you remember from the image they showed at the beginning, I can show that again probably, because here we go. If we look in here, this is what we see on the computer, and this is what you've seen in the video that they've shown as well, right? That was seen there. Mm-hmm. So each one of these is one molecule, and therefore each one of these will contribute towards the final histogram to the same amount. It will just be placed more to the right if it's darker, or more to the left if it's lighter. But if you look at them, if you look at this one, which is high mass, and this one, which is low mass, they are actually the same size and the same shape. They are just different in intensity, which means even if there are a lot of these big ones, they will not necessarily hide the small ones, or the other way around. And that is because what I said before, the measurement is not dependent on the shape of the particle or the size that it has, because the particles are diffraction limited. So what we are looking at here is just the point spread function of every particle, which is the same. So we're literally just counting, each one of these, except that when we take that video that takes 1 minute, instead of a single frame, we will have a lot of frames. And in that 1 minute, we will be counting, a few hundreds to a few thousands of them.
You've talked about how sensitive mass photometry is. What are the concentration ranges that are effective for this kind of instrument and this kind of experiment?
Yeah, it's very low. So I think I mentioned before, we were using typically 10 microliters per measurement. Those 10 microliters are of 100 nanomolar to 100 picomolar solution of particles. So it's quite on the low end. There is a range that we can work with. Obviously, the best amount, or my recommendation to go for if you are working with a new sample and you don't have much information, is to try and aim for the 5 to 20 nanomolar, so that you have some room on both sides to be able to correct if needed. So 10 microliters of say 10 nanomolar sample, it's a very low amount of protein in total that you need for this measurement. That's why you can use it even if you just have a smaller liquid and you don't want to waste it for a bigger analytic or something like that. Yeah.
It's clear that if you don't have enough molecules in your sample, you won't get any data. But what bad thing happens if the sample's too concentrated?
Well, if the sample's too concentrated, what you will see is immediately on the video, instead of seeing those nice 6 or 7 events per frame, you will see a lot of them. Mm-hmm. And when they start to overlap between themselves, then obviously that's when the software will have trouble quantifying them, because the signal of one will affect the other. So that's when you need to decrease the concentration. The software will also give you a warning, basically. So there is a clear metric that tells you, okay, from here on, your data is starting to be affected. It's not too dramatic, but depends how high you go in concentration, you can lose resolution. So the software will warn you for that. The solution is very simple. You just dilute the sample. That's it. So when people come and we do feasibility tests or things like that, I always ask them to bring their sample as they have it in whatever concentration they have it, and the buffer, the same buffer that they have the sample in. so we can check for whether the buffer is clean or not,
Right.
but also just to dilute the sample, and that's really about it.
You've mentioned a lot of the benefits for mass photometry for looking at these kind of protein interactions. Is there anything else you want to add about why mass photometry is good for these kinds of experiments, or when it's best applied?
Well, I think the biggest power of mass photometry is the single molecule capacity, right? So we can talk about many different things, but that is the one that I see having the biggest impact on the people I work with, our customers, our users. And it goes down to seeing every individual population within the sample, which can be an homogeneous, nice population, or not so much. If it's heterogeneous, if for some reason the sample is not as ideal as you would like it to be, you also need to know that. So you will see heterogeneity, homogeneity, you will see different populations. It's not an average measurement, it's not an ensemble measurement, which is what most of us are used to with most techniques. And therefore, it's always good to have at least one single molecule capability in your toolbox, so to say, because you might get surprises. You might learn a lot of things about your samples, your proteins, that you will not learn otherwise. And I think that's the biggest power, and whether that is in the sense of an interaction, of an stoichiometry that was not known, of a protein that is supposed to be a monomer but turns out to phosphate, or of something that was supposed to be something and it's something else. The power of having that information in a single minute, it's really what gives mass photometry the biggest value for people, in my opinion.
Great, thank you. Let's change applications. Can you use mass photometry to analyze micro or nanoplastics, to count them or to understand them?
The short answer would be no. It's an easy one as well. It's not theoretically, yes, you could throw any kind of molecule on this. In practice, though, and this is more of a chemistry thing, but the system is prepared to work with proteins and nucleic acids, mostly. We do work with some lipid protein particles as well, things like nanodiscs and stuff like that work very well as well. That is not to say it's not technically possible. It's just the chemical nature of nanoplastic would be different, so they will typically interact with the measurement surface in a different way. The solvents that are used for those application areas, or things like materials, paints, things like that, will be different. Typically organic, which will require a different setup because they will evaporate, they will dilute the silicon that we use, things like that.
Is it around going back to the plastics question, is it because a plastic is more of a particle and less of a molecule? Could it look at polymer oligomers, but not the plastic particles?
That will depend a lot on the size of the particles. So for proteins, you can look at certain proteins as in polymerization type of thing, or oligomerization of small proteins that form very big oligomers are not that far away from a plastic polymerizing in terms of concept, in a sense. And we do see those. There is a size limitation, though, and I think for anybody thinking about particles, the word particle can mean very different things to different people, right? So when we say we can analyze particles or so, think first about the mass limitations. So the high end is 5 megadaltons. That's for a protein. Obviously, if you're working with something else, you might have a different mass, and the light scatter per mass unit will be a little bit different. But think roughly anything significantly larger than that will be saturating the detector, so will be out of spec, basically, right? There is also a limit of size. I said the way we look at molecules and why they are shape independent or size independent is because they are diffraction limited.
Mm-hmm.
The diffraction limit of the instrument is 180 nanometers, roughly. So anything larger than 180 nanometers will no longer fit that condition. This might happen to some kind of very long DNA molecules, for instance, or maybe a plasmid that is still in the mass range, but it's not supercoiled, might be a little bit longer than that. So just think about that kind of limit. Anything smaller than 5-6 megadaltons and smaller than 180-ish nanometers within the biospace should probably fit. But
Perfect.
anyway, I think if somebody is working with one of these, not an antibody sample, which with everybody knows about antibodies or about proteins or so on. If anybody has something a little bit more specific, feel free to drop a question in the Q&A. We might not get to it today, but we can get to you and have an individual conversation around your specific samples. We have experience with many different things. The first thing, I go to present mass photometry somewhere and I say, we analyze proteins. And the first question I get is, can I analyze something else that is not a protein? So we have a lot of those. We have tried them because we are scientists. We are curious by nature. So we do have a lot of experience with other things and can advise you on whether it might be suitable or not.
You mentioned antibodies. Is mass photometry useful for therapeutic antibody research?
Yeah, definitely. That's a much easier one. Those are a protein, so they will fit within the category. And yeah, that's a very big topic of interest that we also see in our user base, and we even collaborate with some of them quite closely. And again, I think similar as what I said at the beginning, it also applies to antibodies. So there are two ways that you can look at mass photometry and antibody work. The first one is in terms of antibody aggregation, and that's just basically checking the samples, right? Checking for quality of the samples, which at the moment is available on a research space, so to say, but we are aiming soon to also have an offering for GMP environments, so proper QC for antibody aggregation as well, which is something we already do for some viral vectors, for instance. So we are just looking at expanding our offering in there. So when you look, the first example I showed right at the very beginning was an antibody that was aggregating. I can maybe show one that's a little bit more detailed, so to say. This would be this one. And as I said, the single molecule nature of it means that without having to separate to run any column or any chromatography, you can get access to the individual populations. In this example, for instance, we have, it's one antibody, it's the same, it's a monoclonal antibody, but it's been subject to 3 different stress conditions. And we are measuring then, what's the monomer, which would be in the blue area, but also what is the dimer, the trimer potentially, and what's the proportion of this. So here, that is a good example in which the mass is not that important. We already know the mass of the antibody, but we are very, very interested in quantifying that percentage. Mm-hmm. And because it's single molecules, you can really see it goes down to very single-digit percentages. In stress 3, the trimer is 1%. That would be this one here. This one up here, right? And for stress 1 is 2. This one here. So that would be the number of molecules that are under this peak here. Whereas the dimer, for instance, is 8%, and it was only 1% of dimer in the control down here. And each of these measurements, if you just take one, it literally takes 1 minute of recording and maybe another minute for pipetting the sample or so. Obviously, I would recommend to take them in triplicates just to make sure that everything is right and standard lab practices, but it's a very fast way to get this kind of information down to a very granular level. And again, with very little sample and a very low concentration, which is probably covering a different space than other techniques that work at higher concentrations, right? So ultimately, they will give you the full spectrum when you combine them together. And so that's one area of antibodies that we see a lot. And let me show you something else. Apologies for going into the larger view. I don't want to bore you through all of my slides, but this one is also quite interesting. This is the other thing we see a lot of people interested in with antibodies, and is the actual antibody-antigen interaction, right? Can you measure that interaction? And the principle is the same as I explained for the previous antibody DNA nucleosome formation interaction. In this case, it's just an antibody and an antigen, but it's really the same idea. So you will have the antibody and the antigen. We will always recommend to measure them separately as a control. That's what we've done in the top. So for instance, the antibody in this case, we can see it's much cleaner. It's not aggregating, right? There's nothing here. It is degraded to a little extent. This is probably 50 and 100 kilodaltons, so there might be some heavy chains in there, something like that. And then you mix them together and you compare to your controls, right? So you see, in this case, there are two new populations that are emerging. And again, this is what is key of mass photometry. Because it's single molecule, we are seeing these two complexes, the 1-to-1 and the 1-to-2, at the same time in the same tube, without having to separate them, without having to infer this from ensemble measurements or from average measurements. We're literally seeing each population separately in the same measurement. So you can see how first both stoichiometries are actually possible. So, you know, for some cases, this 1-to-2 stoichiometry might not be possible because the antigen is too big. There are sterical impediments, there are other things. That's already a very valuable answer. But then in this case, we're comparing two different ratios of mixing, right? Of antibody antigen mixing. And we can see how the equilibrium moves. We can see how we have more of the 1-to-2 in the second mixing ratio. And you can imagine this being mixing ratios or being different buffer conditions, being different different antibodies themselves, different clones, or slightly different versions of the antigen of the target, anything along those lines. So once you have gone through the discovery and once you have gone through the high throughput screening of thousands of components and you have a few candidates and you need to validate those candidates, you need to characterize those candidates more in depth, I think that's where mass photometry can come in with the single molecule capacity.
Perfect. A lot of people who are working in this space are bound by FDA compliance. Is mass photometry, can it be used with GMP environments?
Yes, that's something we're aware of, and we are working with a few people on that direction. At the moment, it can be used in GMP environments or in environments that require 21 CFR Part 11, basically compliance for the software, for only one application, which is one we have not talked about today. It's a little bit out from the protein world, but it's for adenoviral vectors, AAVs, and the empty and full characterization of empty and full ratios of the viral vectors. That is the first application that Refeyn launched already almost a year ago with compliance package to support in terms of software. The next one we're looking at is most likely antibodies. Don't quote me on that because this is still under a discovery phase, investigation, and so on. But it's something we definitely want to look into because we already have the infrastructure for the software. We just need to, obviously these things need to be developed per application rather than per system. The mass photometry is too wide to develop compliance for that. It would be impossible. So we need to restrict it per application.
Tomás, thank you for your time today. This was a really interesting discussion. I know I learned a lot about mass photometry and how it works and what some of the applications that it can be really successful at. And I hope that those of you who are participating today enjoyed this conversation and learned something as well. And if you want to reach out to Tomás, his contact information is here on the screen. I'm sure that he would welcome your questions and your inquiries going forward. And so hopefully you can find a way to use mass photometry that'll benefit your research in your lab.
Yeah, absolutely. No, thanks, Scott. I think it's been a great conversation. I always enjoy talking about this. It's been my lab for the past few years, so I can really go on and on. So thanks for focusing the conversation on what was important for the people around. And yeah, feel free to reach out at any point to me or in our website.